Okay, imagine trying to solve a jigsaw puzzle, but instead of just one puzzle, you've got thousands, and all the pieces are dumped into one giant box. Oh, and you don't have the pictures on the lids to guide you. That sounds impossible, right? Well, that's pretty much the incredible challenge scientists face every day when they try to read the instruction manuals of life itself. So how on earth do they even begin to solve this genomic jigsaw puzzle?
You know, our planet is just crawling with microbes, bacteria, archaea, viruses, and they are absolutely vital to every ecosystem. But here's the catch. We can't actually grow most of them in a lab. I mean, think about a handful of soil. We can only manage to grow, at best, maybe half of the life that's teeming in there. For the vast majority, we are completely in the dark.
So if we can't isolate them one by one, how can we possibly read their genetic code, their genomes? Well, the answer is to just sequence everything all at once. But that, as you can probably guess, creates a whole new, even crazier kind of puzzle. And this brings us right to the field of metagenomics, which has to grapple with this problem of just immense scale. We're not talking about one puzzle anymore. It's more like a computational haystack filled with countless needles.
The strategy in theory sounds pretty straightforward. You just scoop up a sample of soil, or water, or whatever you're interested in, and you sequence all the DNA inside it. You get the genetic code from every single organism in that community, all mixed together.
So here's where the massive problem kicks in. Our sequencing machines spit out millions of tiny DNA fragments from thousands of different species, all at the same time. The result is just genomic chaos. And our job is to somehow figure out which shredded piece belongs to which instruction manual.
Now, these first little assembled fragments are called contigs. Basically, they're sets of overlapping DNA segments that the software has managed to stitch together. But at this point, they're still just random puzzle pieces. We have no idea which organism each contig actually belongs to.
All right, so how do scientists even begin to sort through this digital mess? Well, the second major step is genome sorting. This is where they use some really powerful computational methods to try and group all those puzzle pieces back where they belong. And this whole process is called binning. The goal here is to sort all of those contigs, all of our puzzle pieces, into different digital bins, where each bin hopefully represents the genome of one single organism. It's literally like trying to sort all those shredded paper strips back into piles for each instruction manual.
But this is incredibly hard to do. I mean, you're dealing with insane complexity from thousands of species. The genomes themselves are often broken and incomplete. Some microbes are super abundant in the sample, others are incredibly rare. And to top it all off, the assembly process itself can make mistakes, creating these chimeric sequences that actually glue pieces from different organisms together by accident.
And a big reason why this is so difficult comes down to the technology. See, most standard tools work by breaking the DNA reads into these tiny fixed-size pieces called kamers. It's a really fast approach for sure, but by chopping everything up like that, you lose a ton of crucial context. And that can lead to the software forcing puzzle pieces together that don't actually fit.
So to get around all these problems, we need a new way of thinking. And that brings us to this really clever workflow called the Jorg method. It was designed specifically to overcome these challenges and assemble truly complete, perfect genomes.
The process itself is actually really elegant. You start with a rough bin from the standard method. Then you use those contigs as bait to fish out all the original DNA reads from the chaos that seem to match them. Next, and this is key, you reassemble only those reads using a much more careful, precise assembly tool. And by repeating this over and over, you extend the genome piece by piece in a process they call digital primer walking.
So the crucial difference here is the tool they're using. Instead of the fast kamer method that just shreds all the data, Jorg uses a tool called Mira. Now, Mira doesn't chop up the reads. No, it carefully compares the entire read to find genuine overlaps. Yeah, it's slower, but it maintains what's called read coherence, which just means it makes sure the puzzle pieces are actually consistent with the original data, preventing all those false connections.
So what happens when you apply this much more rigorous process? Well, the Jorg method gives you 3 very distinct outcomes, and each one tells you something really important about the quality of your genome puzzle. It can either circularize, stall, or shatter.
Okay, the best possible outcome is circularization. Since most bacteria have circular genomes, getting one single, long contig where the two ends overlap perfectly is the gold standard. It's basically definitive proof that you've solved the puzzle. You've got a complete, high-quality genome. Mission accomplished.
The second outcome is idempotence, which is just a really fancy word for stalling out. The process just hits a dead end, and it can't extend the contigs any further. This tells you the puzzle is probably incomplete, maybe because of a tricky repeated region or just a gap in the data.
And then you have the most dramatic outcome: chaos. The whole bin just shatters into a ton of tiny little pieces. But get this isn't a failure of the method, it's a huge success. It means the original assembly was totally wrong. The first puzzle pieces were glued together incorrectly, and Mira's strict rules just broke them all apart, revealing the hidden mistake. It's like having a built-in quality control check.
So why go through all this trouble? Why is this so important? Because with a complete circularized genome, you can finally read the instruction manual clearly. And that's when you can uncover biological secrets that are completely invisible when you're just looking at fragmented data. Get this. Using this method, one single study assembled 36 new, complete, circular bacterial genomes from data sets that were already publicly available. Just let that sink in for a second. That's 36 perfectly solved puzzles that were basically hidden in plain sight this whole time. And these weren't just any old bacteria. They all belong to this massive, super enigmatic group of microbes called the candidate phyla radiation, or CPR. At the time, only about 40 other circular genomes for this entire group had ever been published. So this one study nearly doubled that number, giving us a massive new window into this microbial dark matter. And this led to the first really big discovery. In almost all bacteria we know of, the genes for making ribosomes, the cell's protein factories, are all packaged neatly together in a row. But in these complete CPR genomes, those same essential genes are scattered all over the place. Sometimes they're even on opposite strands of the DNA. This is a fundamentally different way of organizing life's rulebook, and it's something you could never have seen in fragmented data.
Okay, the second discovery is, well, it's even more mind-bending. They found these totally bizarre versions of an essential gene called RNase-P RNA. In one case, a specific structure within it, a structure that's considered one of the most conserved across the entire tree of life, was just gone. It's like finding a car that runs perfectly fine but is completely missing a critical piece of its engine. It shouldn't work, but it does.
And that really brings us to the key takeaway, and it's captured perfectly in this quote from the researchers themselves. You can only make these kinds of huge paradigm-shifting discoveries when you are absolutely confident that your data is complete and correct. You have to solve the puzzle first.
What this work shows us is that with the right tools, we can finally begin to piece together the instruction manuals for the vast, unseen majority of life on this planet. And it leaves us with a really thrilling question. As we get better and better at solving these genomic jigsaw puzzles, what other fundamental rules of biology are we going to discover were actually meant to be broken?